if there are any questions, we can start just while people are coming back. But I have got something new, brand new to tell you about the human hippocampus in a translational thing before I move on to the next topic. So, are there any questions? Okay. I think it's just pattern separation of whatever inputs you have. So whatever came in is set up to be more orthogonal, more sparse. It's the sparsest bit of the brain, as far as I know, when people record in rats, okay, for its representation. So yeah, nothing sort of special. Which reminds me sort of about something. So what's going on in the hippocampus? Is it a representation of the rich sensory world? What is it? Well, what I was showing you is that the hippocampus has to learn how to retrieve the information back in the neocortex, with the rich information back in neocortex. So in a sense, we can say that the hippocampus is pointing to the correct place at which to retrieve the neocortical representations. So that's a little bit like saying, it's a bit like the old index theory, but it's not really a magical pointer that couldn't work in the brain. It's a well-formulated theory of how you would do retrieval back to cortex, okay? So that's a comment on. Any other things? Okay, I'm going to just tell you then something quickly new about the hippocampus. So basically, there are lots of memory systems in the brain. Another favourite one of Simon's and mine is the one concerned with emotion, reward, decision-making in the orbitofrontal. Ooh, what's happening here? Oh yeah, that's right. In the orbitofrontal cortex. What I want to tell you is how the hippocampus is related to this system. One thing we know is that very important in emotion are rewards and punishers, and particularly non-rewards. If I don't get an expected reward, I can be angry or whatever, or sad, and sad leads to depression. So the bit of the brain that we believe implements this type of memory, object to reward or punishment, is the orbitofrontal cortex, and the evidence for that is a cell recorded by Simon Thorpe, redrawn here, Simon, beautifully, neatly, which does the following. This is a monkey doing a visual discrimination task. A visual stimulus appears at time 0. He licks expecting reward, but doesn't get it because we've reversed the reward contingency. This neuron fires for about 10 seconds. It's still firing at the start of the next trial, only to not obtaining the reward you expect, the sort of thing that would make a human sad, okay? This is negative reward prediction error, in fact, okay? So we discovered that in orbitofrontal cortex. We then, recently this year, did the first voxel level functional connectivity study of depression with 900 people imaged in resting state fMRI. What you're doing here is you're looking without a task at the correlations between every pair of voxels in the brain. The number of pairs of voxels in the brain, if they're 3 millimeters by 3 millimeters, is that number of pairs. So you have to be a little bit careful about your statistical correction with Bonferroni, which is, or FDR, which is why we had 900 people in this study, okay? And so we were able to get, as long as we had functional connectivities in depressed people, which were different, at p less than 10 to the minus 7, they were significant. And this was a big study performed in China where you can do, where you can get these enormous numbers of patients. So here's what we found. These are coronal slices showing the voxels in the brain that have altered functional connectivity in depression compared to controls. Look at this. Medial orbitofrontal cortex is featuring, but so here is lateral orbitofrontal cortex. Lateral orbitofrontal cortex is where non-reward is represented in humans, okay? Medial orbitofrontal cortex is where rewards are represented in humans. But look, what's this bit of the brain? Can anyone tell me? Who knows? Does anyone know what that is? It's perirhinal and entorhinal cortex, Okay? So there are lots of voxels in the gateway into the hippocampus, which are different in depression. So we then sort of make these correlation matrices. These are brain regions, and we can say that there's decreased connectivity here between, for example, parahippocampal gyrus, blue, is decrease, and medial orbitofrontal cortex. And if we look at lateral orbitofrontal cortex in this column, it has increased functional connectivity with precuneus, angular gyrus, and the inferior temporal cortex. So here's a summary. What we discovered is that depressed people have decreased functional connectivity, decreased correlations between the medial orbitofrontal cortex reward pleasure system and the medial temporal lobe memory system. What does that mean? Well, the interpretation we suggested in that paper is that your memory system is processing fewer happy memories, you're biased towards sad ones. And then lots of people wrote into us as soon as we published the paper and said, yes, that's what it feels like. So that's just a link into translational research and letting you know just what's happening now. The other thing I want to tell you is that lateral orbitofrontal cortex here, the non-reward bit, has over-connectivity with precuneus, which is where the sense of self is represented in the brain. So we think this is related to too much negative sense of self, low self-esteem, which always happens in depressed people as they're going into a period of depression. And then angular gyrus left, language. So we think this is thinking about negative thoughts. And then this is inferior temporal cortex being interpreted negatively when you see stimuli, okay? So that's just to say that the medial temporal lobe is of interest in relation to depression and the whole hippocampal system, but because it's biased, we think, towards unpleasant events. Okay, so that's just to say here's a bit of translational stuff. The hippocampus is interesting even in relation to depression. Okay, so now I want to go on to a difficult issue. Memory's sort of simple. All you do is store something and retrieve it. The real problem to understand in the cortex is how it computes something new. How do we ever compute a representation that responds to something like this, which you can recognize from different viewpoints? That's incredibly difficult when you see this, okay? So what's going on here? That's what I want to talk about. And in this work, some of the key people were included Nick Agalopoulos, who helped a lot with that. And then Gustavo Dicco did some modeling with us. Right. Okay, visual system. Everyone knows Ungerleider 1995, V1, V2, ventral visual stream, V4 to inferotemporal cortex, dorsal visual stream coming up here, V3 to parietal, here it is, coming up here. I'm going to talk about this stream down here. Notice that this very nice ventral stream does come up into the hippocampus, but the ventral stream is also connecting into orbitofrontal cortex. Okay, so it provides the what input. So how do we build interesting representations from V1 down through these multiple stages to inferior temporal visual cortex? And what is the coding? So I was tempted to put this in because Simon has an interest in this issue. So here was basically the discovery of face cells in the brain. We were presenting stimuli, here we are, time zero, and if we present one person's face, you get a big neuronal response. Other faces, not much. Non-face stimuli, not much. These neurons have interesting properties, they're invariant. So here we are, transform invariants for retinal size. This is the size of the face subtended at the retina. The neuronal response is essentially independent until it gets down to be about 1.5 degrees across, the size of a thumb at arm's distance, okay? So typically we find that they're invariant with respect to size. They're translation invariant too. So if I present my face here or center it 8 degrees away here, I get exactly the same response. And if it's a different face, no response anywhere. So they're translation invariant. Absolutely crucial for recognizing something. You can recognize this if it's on this bit of your retina or that bit or that, okay? So that's what we discovered about face cells. Some of them have transform invariants, okay? So here we are, single neuron with different views of a head. This is the back, this is the front, left and right. This is one face, a high firing rate. This is firing rate plotted out there. High firing rate to one head, almost independently of view, and a different head, lower firing rate. Okay, so some of the neurons, not all of them. Here's an example of a neuron that's not view invariant. This one responds to the profile view, side view like this, but not a front view as much, okay? So some of them are transform invariant, and we showed exactly the same for objects. So the monkey lived with this bunch of objects in his home cage for a bit, so he could learn to recognize them from different views. And then we showed in the experiment, when recording from a single neuron, all these views of an object and all of the objects, and the neurons distinguish. So here's one object with a high firing rate, another object with a lower firing rate, another object with a lower firing rate. So this is true for objects and faces. Now, while we were exchanging views about how wonderful neurons are, here's one of the most wonderful neurons I've seen, which again is informative so that you know what can be encoded in the brain. Here's a neuron that's doing object-centered encoding of faces and heads. So what we're doing is, in this case, we get a high firing rate if we have ventral flexion. That is, if the head does this. This is related to social behavior. If the head does this, dorsal flexion, here, there's no response. But the point is, this happens for frontal view, profile views, even for the back of the head. But here's the weird thing. We then turn the monitor upside down. So this, at the moment, is going downwards across your retina, if you like. When we invert the monitor, it's going upwards. But the object-based description is the same. Your head's bending on your body. The neuron is coding in object-centered coordinates because it still responds to ventral flexion, even when it's upside down, okay? So these neurons are in the cortex in the superior temporal sulcus, and I'll show you computationally how we think they're being produced. You also get some neurons that code in the STS for face expression. This is a threat, mild threat, and a calm face, and this neuron's responding to calm and less to threat, for example. So there are expression neurons. Now, here's just a little bit about coding. We use a large bunch of stimuli, measure the firing rate to all of them. And the question is, how is information encoded? Here we are, one stimulus, fourth stimulus. Looks like firing rate might have something to do with it. So we tested that. What you saw is that this is a fairly distributed representation. This is the firing rate going up to 100 spikes per second for a single neuron. This is 68 visual stimuli, many of which were faces. So the best response is about 90 to the most effective face, second one about 80, then 70, then we're coming down to about 50 and so on. So we have this typical profile for most inferotemporal cortex neurons. We can put that into a more useful form mathematically by plotting the probability of having a particular firing rate. We see a high probability of low firing rates and an exponential decreasing probability of high firing rates. That's the code, roughly, that's being used. Given that this fits an exponential distribution rather well, that sets technically the sparseness by our measure to be 0.5, okay? This data was obtained while the monkey was just looking at a screen with a movie, okay? So it had a wide range of statistics. We get exactly the same shape, I don't know if I have a slide here, yeah, the same shape, when we just measure the probability distribution of firing rates to single stimuli presented, but over a very large number, okay? So that's absolutely typical for inferior temporal visual cortex. They're not grandmother cells as we find them. They tend to have this exponential firing rate distribution. And then if we look at the sparseness here, in which the sparseness A is measured by effectively this firing rate to stimulus I divided by the firing rate to the whole set. Now, here's the sparseness distribution with that particular measure. You can see if it was totally sparse, a grandmother cell, all of the measures would be down here, but they're not. They're much higher than that. Okay? Now, how's information represented in your brain? Well, we applied Shannon information theory to the coding, and you find that for these cells, you get maybe about 0.4 bits of information if you listen in for 400 milliseconds. And if you listen in for 50 or even 20 milliseconds, you have enough information to tell you what object it is. So this is crucial to understanding how the cortex works. I can read out from any one area of cortex in about 20 milliseconds, in fact, all I need to know for the computation to do at the next stage. This enables everything to ripple through quickly. Okay? Yes? Single neurons. That's single neurons.
Just measure the activity of a single neuron
yes.
in a single trial.
Yes, and I get so much, that's right, and I measure how much information I have about which of a set of 20 stimuli or 60 stimuli, okay? Right, and so these are just the conclusions that a lot of the information's available in a short term. So Heim is now thinking ahead and saying, what happens if I have lots of cells? How can I read the code? So we developed a new way of applying Shannon information theory, which just says the following. Measure the firing rate, which might be an increase from cell 4 to stimulus 1 and a decrease from cell 1, and same for stimulus 2 across our population cells, the same for stimulus 3. And then we decode. So we take a single trial of data, crucial, as Heim says, and we find this profile of firing. Can anyone tell me which stimulus was shown on this trial? Any guesses? Yeah, I think it's one. Yeah, it's one. It doesn't match any of them perfectly because each trial is slightly random. So we measure the mutual information between our presented stimulus and then our population averages, okay? And that tells us how much information we get from all of these cells about which stimulus was presented on a single trial. And so we can ask this question for different numbers of cells. I'm not going to go into the details of how one does the measurement, but here's what happens. This is information in bits. This is the number of cells that you include when you're, say, presenting 20 stimuli, I think this was. And you measure the information goes up like this, almost linearly. If we had had more stimuli, it would have gone up linearly. That is, for 20 stimuli, you have just over 4 bits of information. So what this is doing is asymptoting to 4. What this tells us is something very interesting about cortical coding. Because the information goes up linearly, the neurons are effectively coding independently. You add together the information. The information is a log measure, so that means that as you increase the number of cells, the number of stimuli you encode goes up exponentially, and this also shows that you can read the code pretty well with dot product decoding. That's the simplest form of decoding a neuron could do. Measure the firing rates of all its inputs, and through its weighted synapses produce firing. Okay, so this works with biologically plausible decoding. So that's part of the power of having a distributed representation, that you can code so much with relatively few cells. The number of stimuli you code goes up exponentially in the number of neurons. Yes?
So the data is from single unit recordings?
Correct. No, not entirely. No. We measure simultaneously from neurons, okay? And that's just to say dot product decoding, which perhaps most people know. But what happens then if you record from cells simultaneously? Now, it's possible that, for example, if I have cell 1 and cell 2, and they're temporally synchronized in some way, I could tell it stimulus 1 And if those two cells are not correlated with each other, cross-correlated, then I might know it's stimulus 2. So potentially, I could code information in the correlations between cells. So we set up our algorithm to take into account not only firing rates, but also all the correlations between cells, and then reran the analysis with simultaneously recorded cells, okay, usually in small groups of 4 or 5. And here's what we found. Total information you get from your ensemble is there. If you just measure it from the number of spikes you count, you get almost all of it. If you look at the cross-correlation firing that's stimulus dependent, you get very little information. So the rule is almost all the information is in the spike counts in inferotemporal. Almost none of it is in the cross-correlations. So Wolf-Singer had a good hypothesis, but it's not supported by what happens empirically. Even when you do a search task like this, we asked our monkeys to look for one of the objects. I don't know, I think that's a hand. And if he touched the hand, he would get fruit juice. And he had to do it then under real visual conditions and find the hand in this scene, just to make sure that all this correlation stuff that we had found would apply to natural vision. And there is a hand somewhere in there. I can't see it at this scale. But here's what we found. Even under natural visual conditions, the cross-correlation information, the temporal binding, if you like, is very low, and the rate information accounts for almost all of the information, okay? So that's just to say that information encoding, even in natural scenes, has little stimulus-dependent cross-correlation. Most of it's in the spike counts, okay? So just a rate code is basically the conclusion that comes out of multiple cell information analysis. Now, just because we have some very quantitative theoreticians here, I wouldn't claim that the neurons are independent for very large numbers of neurons. We've done it up to about 50 neurons, and it's more or less independent. But if I went up to sort of 1,000 neurons, I wouldn't argue it'll go on forever. Otherwise, you get absurd amounts of information that you can read out, okay? So I'm going to skip that bit, I think. Right, okay. Look, that's what's computed in the inferior visual pathway. A beautiful representation that's ideal for a different memory system. For example, for the reward, the object and reward memory system. Why is it so good? This is to tell you why you form these representations. So look at me now, okay? I'm going to smile at you. That's great. So I'm a nice person. What happens if I show you this?
You are nice now.
Very good, very good. I compliment you on that. So basically, what's happening there is that you were intended to generalize and say, I'm still nice, okay, like this. In other words, all you need is an object representation from your inferotemporal cortex, okay? And then if you send it to your orbitofrontal cortex, once you've learned about one instance, it generalizes to all of the others. Absolutely crucial, okay? That's why we form invariant representations. The same applies for the hippocampus. You have to have an invariant representation of a person if you're going to do an object-place association, okay? Right, so how is it computed? Okay, so let's firstly look at what happens. So here we are, primary visual cortex, secondary, V4, posterior inferotemporal, anterior inferotemporal. What happens is you have convergence in the forward pathways. A small bit of cortex projects forward into this cortical stage, and it happens stage after stage. So the first thing to notice is by the time you're at the end, you can potentially look over a large part of space for where something was represented. That's part of what's needed for translation invariance, okay? Now, the other thing to notice is the receptive fields increase in size as you go through by a factor that I think something like, I don't know, 2 or something, 2.6, I can't remember offhand, okay? So that's the hierarchy, and then the latencies here allow you about 20 milliseconds per stage. Those are the constraints, the anatomical and temporal constraints. So if you make a model of this, it would have a single neuron here that receives from a small part of the preceding area, and that's repeated over and over again. Can we get this architecture to do anything interesting? Potentially, a single neuron here could respond to an object anywhere in the visual field, but you have to have the right synaptic weights set up in the forward pathways. How on earth do you learn that? That's a massively different problem. That's what all these Google nets, deep nets, are trying to do nowadays. And the latest is they go up to 100 layers to try to solve this problem. How does the brain do it? In 4 layers. And the brain can't use more than 4 layers because otherwise it would run out of time. It would take it too long, okay? Incidentally, I asked, for those of you who are into machine learning, Jan LeCun about why he put in 100 layers or even 1,000 layers to the deep net. And he says, oh, I have no idea why it works, and we had no prior hypothesis. We just tried it and it worked better. Okay. So it's not driven by theory, it's just driven by what works, okay? A little better. So how could such an architecture do something useful for you? So let's start off with V1, where we have Gabor filters or simple edge things. The crucial stage is put together feature combinations, as Hubel and Wiesel said, and form neurons at the next stage, V2 perhaps, that respond to a combination of that and that, but in the correct relative spatial positions. Then I can code for that as being separate to that. And that's the key for binding in your cortex. You bind by forming feature combination neurons, not by using temporal codes or something, as I just showed you. The temporal codes don't seem to be being used. So I have to do this feature binding early on. Otherwise, if I tried to do it late on, I would never know what the relationship is between one feature and another. So I have to do it where I have a precise retinotopic map. And so I form this in, say, V2, and then what happens is if I form combinations of this here, I could form something that looks a bit like that object, and for another bit of the retina, I would do the same, and I've got to map then this combination to the same neuron here. How do I achieve that mapping? That's the interesting question. Okay, so we made a model which is basically called VisNet and which has 4 layers. And what it is a series of hierarchical computer networks with local inhibition, convergent connections to each neuron, and Hebb-like plasticity with an amazing twist. So here's a competitive net. I've more or less shown you how that works. Provide a set of inputs, random connections. This neuron might fire a bit faster than the others. It then wins and turns off the others, so you learn better that this set of inputs activates that. This is not a bad way of doing categorization. It's not perfect, but it works moderately well. And as I say, there's software to demonstrate this to you. Okay, so then in VisNet, we just have 4 layers of competitive net. But this is our trick. We use the statistics of the real world to act as the sort of trainer for the system. There's no supervision anywhere in cortex. You never have a tutor, as you do in a deep layer net, saying this neuron has to code for one person and this neuron has to code for another. So it's all got to self-organize without a teacher. But we're saying there is a sort of teacher which is the world. So if you look at my hand now, it might transform a bit and move over your retina. So there's something, if you could learn from the different views represented in the short term, second or two, then you might learn that that is the same as that. The same with my head. In the short term, you could potentially learn that all these different images are really part of the same object, and that's all we do, okay? So what we're arguing is that having a slightly slow feature to our learning that lasts for perhaps a second would help you to capture all of the invariances that could be captured at any one stage of the hierarchy. And so here formally is the learning rule The change of synaptic rate is proportional to the presynaptic firing rate multiplied by the average of the postsynaptic firing rate over a short time period of about a second, okay, in a sense. Okay, so that's all it is, slightly modified Hebb rule. How could that be implemented? Well, you've got attractor networks in neocortex that tend to keep the firing going for at least 100 milliseconds. We've shown that. But also the NMDA receptor has a time constant of 100 milliseconds, which is nice. Okay, that helps you to smooth over time. So we then present Gabor-filtered images to the input. That's our simulation of V1. We take real objects. Here are some simple ones, but these are interesting because these are different combinations of two lines where you can only recognize the difference by doing some form of feature encoding. So we find out if our model can learn to represent each of these things with different neurons in a transform invariant way. So here we are. This was our very first experiment, I think. Different locations for these objects. And here we find a high firing rate in the last stage of the model to across invariantly with respect to where it is, but not to the other two stimuli. Okay, so the model can use time. And we only see this good discrimination if we use the trace short-term memory modified Hebb rule, not if we use a standard Hebb rule without the temporal trace. So the temporal trace is crucial. And because we use realistic filtering, Gabor filters, just like V1, we can present real images to our network. And what we do is we would present views, say, of this person together in temporal sequence, and then we would present views of this person and of me, okay? And the network, because it sees the different transforms, can learn to associate, say, that with that, okay? So that's how it's trained. And then here, what happens is we have a cell that's responding in the fourth layer of ISNet to one of these faces independently of view, but not to the other face, okay? So again, it can do the right thing. So the temporal code is one trick that the brain uses. Another trick is spatial continuity. So just think of this object. If it transforms quite slowly, like this, then there's overlap of the spatial representations. Do you see? So this is pretty similar to that. It turns out that just spatial continuity can also be useful to learn invariant representations. Here's the intuitive idea. If I have these three inputs active, and they happen to be activating this neuron because of the connectivity that's present, then if I slide my stimulus to this position, then two of those inputs will still activate the neuron, but the third input then can become Hebb associated. So even without temporal delays, I can learn a bit. And we did a big experiment with lots of these shapes, which were transforming by relatively small angles without a trace rule. And the system can learn, I won't bother to show you the actual data from it, but basically the system can learn just with a trace rule. The important thing to understand about this then is that continuous transform learning tends to work well with these small time steps. But objects show what we call catastrophic changes as I alter the view. So if I get this, Simon can tell what's going to happen. if I take this object and turn it like this until it's horizontal towards you, there's a massive change in what it looks like on your retina. And in the field, that's called a catastrophic change of view, and it would be. Okay? So the point is, spatial continuity won't do for that. So that's the situation in which the temporal code comes in as being particularly important, because then I could associate this view, because of time, with something that looks totally different. So these two things are slightly complementary. The trace works with large steps and with very sudden and major transforms of the image, okay? So, in this network, is feature binding important, or is it temporal coding? We did a big study in which the neuron was given either a single thing or a combination of 4 edges. And all of these are overlapping with each other a lot. The correlations between them are high. And we asked our network to learn to discriminate all of these. You see this object from this object, and this object, and this object, and that object, and that object. And it can only do that if it's forming feature combinations. And basically the system learned that very well, okay? So it can separate out objects which are pretty similar to each other because it's using competitive learning to do feature binding, okay? So here's another funny twist that comes out from this. So this is all ventral visual system. Could anything like this work in the dorsal visual system or in some other bits of the brain? So look, here's a weird thing. In the dorsal visual system, you might have a rotating wheel, okay? And, you know, it's like in these cowboy movies. You see something going around like this, okay? But look, what happens when the wheel shifts across your retina? The local motion would be down here, but up here. So you can't do it on local motion. If an object's moving like this, and it goes along here, you have to do an object-based representation of motion. Now, there's some evidence that, well, there's good evidence, that's computed in the dorsal visual system. The neurons actually can respond to global motion of objects like that. So how does it work? Well, we think it's just the same as VisNet. So we have an input layer which now doesn't represent edges, but it represents local motion, exactly what's represented early on. Small movement of a degree or two across the retina, okay? So if you think of putting together some motion signals here, in an early stage, you would get global planar motion. A movement, for example, like that might activate a neuron, okay? But if I then put together that and that, up and down, I can form a neuron here at MSTD, which responds to rotational motion, but for one location in space, and for another location in space here. Do you see? It's the VisNet principle of slow learning. And then if I put together in a next layer, MSTD or higher, this and this, I can do this invariantly with respect to position. So we did that in the model, implemented it, and here's what happened. So in that model, we were able to get these neurons, this is the number of cells, this is the information, that if we train with a trace rule, can learn this, and if we just use random connectivity, won't. So you can do global motion. Now look, here's this interesting cell I showed you. Could the model of global motion in the dorsal system learn this? Do we even do a transform invariant global motion thing? So we tried it. We set up cylinders that were moving like this, but they were dark here and light here, so it was object-based, because then we could take the cylinder and show it inverted, and then the local motion is opposite, even though it's the same object-based motion, okay? And so when we do that, we find the system, as long as it's trained with these things in sequence, slowly inverting, can learn this object-based motion perfectly well. So that's saying that some of what goes on in the dorsal system may use the same computational principles as those in the ventral system, not everything. So here's what we want to say about invariant visual object recognition, that you can use trace rules and you can also use continuous learning to help you learn it. And the most recent description is in that book. Okay, so that's really what we want to say then about vision, that now a really complicated problem, invariant object recognition, can be solved by networks that we can more or less understand. okay? And then just to say, what's really crucial about what's going on here, let me compare our model with Reason, Huber and Poggio's Max model of invariant object recognition. So in their model, what you do is you start off with effectively simple cells, and then you just do a sort of analysis of those in which they're connected together to form complex cells, but without any learning. You're not actually learning feature combinations in this system. They just have a series of filters, and they try to implement complex to simple cell transforms by having cells here that would respond a little bit to several simple cells. They just do a pooling from simple to complex, but nothing more. And then eventually they get through to things that they claim are view-tuned cells. But there's no learning. So how does their system perform compared to ours? So here's one key thing that you have to solve. You can take real objects and transform them and see whether you have learned that the views are the same. And so VisNet can do the invariance learning. This is the percent correct, and this is the number of training exemplars that we're learning about. HMax can't do that. The reason is it has no learning in it. It's got no way of knowing that that is the same as that, you see. All it is, effectively, is a feature-based analysis, okay? So here's another key property of what you have to be able to do. So you can recognize this individual independently of all those transforms. And after you scramble the system like that, inferotemporal cortex neurons don't respond to the scrambles. And you probably don't either, terribly well. You don't know who it is, okay? So what does VisNet do to that? So VisNet does this. It responds very well to the normal, but not to the scrambled, because it's learned feature combinations using its competitive learning. It has to have the parts in the correct spatial location. But HMax fails miserably because it's got no way of associating things together, okay? So again, that shows you just the sort of property that it's key to understand. And then the sort of last example is, say I want to distinguish these two cups. So this cup looks like this from one view, and it's got the person's name at the bottom of it, and that's it from another view. And if you look at this for a bit, you'll probably recognize this cup belongs to Bill. If you look at this beaker, you'll probably learn it belongs to Jane. Okay? So how does VisNet do on that? Well, when you transform this, it of course associates together all of those views and does perfectly well. And here it is doing well. On the Bill exemplars, the different transforms, but not on the Jane exemplars. But if we look at HMax, it can't do that because it's never associated together things like the words with the shape. So it fails miserably, or because it's just a feature analyzer. It just responds, in fact, to the words. So if any image has words in it, it says, oh, that's different from things that don't. So can you see? I'm trying to give you an idea of really what's going on when we do interesting computation in our ventral visual system. There are a whole set of principles here, okay, which are set out in that book. Okay, here's my last slide. So there'll be a few minutes for discussion of either hippocampus or this, what I've been saying here. And I think I will stimulate a bit of discussion because this is my view of cortical coding. And you've heard Simon's view, and I'm sure he'll talk about his view a bit more at any time. You might even ask some questions. So cortical coding, very sparse distributed coding in the hippocampus to maximize the storage capacity. So concept cells. What are concept cells doing? So I think concept cells are just this. They're just the sparse representation of stuff that's come in from places like inferotemporal cortex. So it goes, because it's a sparse representation, with a less distributed coding than's present in inferotemporal cortex. And why? Because that's good for memory, to to have a sparse representation. So that's what I think a concept cell is. And of course, if the cortex can code for the sight of someone and the sound of their voice, which probably is going on in superior temporal gyrus, then the representation in hippocampus would represent both the person as seen and as heard. Okay? Right. So I told you that the sparseness in the monkey is more sparseness than our measured values because we used only one room. So sparse distributed representations with an exponential firing rate distribution are used to maximize the information representation capacity. Okay? So what I showed you in this lecture is that the information goes up the number of stimuli goes up exponentially with the number of neurons. That means that all a neuron has to do at the next stage of processing is to listen in through its random forward connections to a relatively small number of neurons in the preceding stage, chosen at random, and it will still have a lot of information about what object was present. That will never be true of grandmother cells. If I have a grandmother cell that's coded for one thing, then if I want something at my next stage to do something sensible, it would have to listen into lots of grandmother cells. Okay? So that's fairly key, that these sparse distributed representations are used for the rich description of everyone's sort of clothes, face, hair, eye separation, and all of those things. But it's just made into a reduced description, a concept, if you like. Now, let me say something else about concept cells for Rodrigo. So when we have object-place combination cells in the hippocampus, we think they're the same as concept cells. Okay? So they respond if I show an object and I can retrieve the place, or if I present the place and they retrieve the object. So in a sense, it's just a one-shot memory that does these simple associations that can therefore learn to respond to combinations of things. So the concept cell, I think, is a bit like that. It can respond to the different properties, some of which have been brought together before you get into the hippocampus, and some of which are crucially learned in the hippocampus. Okay, right. Information representation is high because the neurons have this independent information encoding. And competitive networks help you to form independent representations at the mechanism level. In other words, we're approaching getting down to even understanding how it's produced. That's the point of that book over there. The code can be read in a biologically plausible way. Now, sparse distributed representations allow for generalization and completion. So if I have a sparse distributed representation, several neurons will be active for that. If I present only a subset of the information, then some of the neurons will be active, and I'll still be conveying some information. And then in structures that do generalization, such as competitive nets, pattern associators, and auto-associators, you can retrieve almost the whole representation from any part. That's not a property of grandmother cells. So that's a major advantage of having this sparse distributed representation. And oh yes, and I've said this thing about those. And there are multimodal concept cells in other bits of the brain. So they're not unique to somewhere like the hippocampus. So as Simon was telling you, when you go into places like orbitofrontal cortex, you get neurons that are sort of concept cells because they respond to the sight of, say, a food, the smell of a food, and the taste of it. So they're sort of multimodal things. And any one of those inputs, here's the key point, would raise the concept, this is a banana, if you want to call it a concept. But I would just call it, to make myself clear, if it's very sparsely represented, then it's like what you call a concept cell, but I would just say it's a sparse representation. That's useful for some things. Now, in orbitofrontal cortex, a sparse representation can be useful for one of my favourite things, sensory-specific satiety. If I have a lot of one reward, my neurons active for that reward adapt, and I no longer like that reward as much, but I still like other rewards. So the point is, these quite specifically tuned neurons can be useful for different functions, and one is this reward-specific decrease that you get sensory-specific satiety in orbitofrontal cortex. So what I'd like to say is, in a sense, concept cells are not unique to the hippocampus or to that type of memory system. They're used in other memory systems, including the emotion one, for object-to-reward associations. Okay, 5 minutes for questions. Any questions?